All right, so in this stream, we're just going to be finishing practice problems and then have an open question time. And like I said, if this is only 30 minutes, that's fine with me. I know you all have a lot of homework and other really hard classes, so we'll just kind of go over the basics. All right, this is kind of to finish up yesterday when someone asked about semi-conservative replication. I drew a really terrible picture because I do not yet have a tablet situation where I can draw on my screen. So here's a nice picture of semi-conservative replication for those of you who are asking. This is one of those key characteristics of one of the nucleic acids, DNA. So we know it's a double-stranded molecule, and semi-conservative replication is just a way to describe DNA replication. It's where we've classified DNA replication as semi-conservative. So conservative as in how much of the original strand is it keeping? You think of someone who's like conservative with their money, they don't spend a lot of it, they hold on to it. And so if this was like fully conservative replication, that original parent strand would hold on to itself and be identical beginning to end. And so it wouldn't, it would rejoin the parent strands after replication. But you can see here with the blue and the pink, the original parent strand is double-stranded with blue. And then that first generation daughter strand, you can ignore the second generation, but this first generation is what is important. It's half and half new, half old. And so it's semi-conservative. It still has all the same information. It's still identical. But as far as, you know, if the original parent strand is going to be semi-conservative. So it's half parent, half original parent. Okay, and then I had another person ask about functional groups, and we didn't get to that yesterday either. So here's a picture of a molecule, and it's got a bunch of different functional groups in it. So just go ahead and take a look and see if, you know, some of them are more obvious than others, like the phosphate functional group, which plays a really key role in energy transferring. If you could describe what this functional group looks like in this diagram, go ahead and type that in the chat box. So which one is the phosphate group? You can feel free to use colors or names of elements. So which one would be the phosphate group? Just type that in. Let me know which one is the phosphate functional group. Top left. Yes, Sandra, absolutely. Good. So yeah, the top left, your yellow phosphate kind of looks like a tennis ball, and then it has 4 oxygens. Phosphates in general as a functional group play a huge role in energy transferring. So I know a few of you yesterday said that you were in cellular respiration already. And so once you really get into that, and we'll talk about the molecule ATP, it's adenosine triphosphate. So it's a molecule that actually has 3 phosphates next to each other. It's a super high energy molecule. And so when we're able to break one phosphate off, it is actually a huge release of energy. And that energy is used by the cell to do all sorts of things, to bring things in, to take things out, break things down, all sorts of stuff. So phosphate is kind of an energy transferring group. Let's look for if you can find the carboxyl group, or your teacher might have called it carboxylic acid. And so if we're looking at a carboxylic acid, tell me where you see that. A carboxylic acid is something, as an acid, it's going to be able to unload hydrogen ions into solution if needed. So carboxylic acid. And if you guys haven't done this, let me know. If you did not cover functional groups in Unit 1, let me know. It's usually something I try to cover before going into macromolecules, but it just never made it into any of our streams. The middle-ish part. So if you're looking at this middle one, can you guys see my cursor going over this? Javier, so we're talking about functional groups. Somebody had a question about them last night. There's several groups. They're basically, they're called functional groups because they are common molecules that are found in biological organisms, and they have a job. So that's why they're called functional. So for example, like phosphates, they're seen everywhere, and their function is to help with energy transfer. Where like a carboxylic acid, its job as an acid is to be able to unload or take, mostly unload hydrogen ions. So it's actually this one far to the right where it has a carbon double bonded to an oxygen, and then there's an OH hanging off on the end. So it's the far right molecule. It's a carboxylic acid. That's also a part of your, you see that in your amino acids when we talk about proteins. On the other side of this molecule is where we see the amine functional group. So we have, I'm going to type the words in here. So we have the amine group. We have a carboxyl group. Oops, I typo group. Phosphate, carbonyl. So a lot of these groups, and hydroxyl, those are I would say those are your 5 main functional groups that are seen throughout. So the amine group right here can act as a base where it can pick up hydrogen ions, right? So it can actually hold on to extra hydrogen ions when needed to help stabilize an environment. If something is way too acidic, it can pick up hydrogen ions. And help with pH levels, because things like in your blood, for example, if your blood were to become too acidic or too basic, it could be fatal. So these molecules that are floating around, they have certain functional groups that can help out. The other one, their hydroxyl group would be just an OH right here at the bottom. And then the final group is a carbonyl group, which is just a carbon double bonded to an oxygen. This topic of functional groups is not something that's explicitly stated in the new AP Biology curriculum, but it is something that it's worth going over, and especially since someone had a question yesterday. I know she's not here tonight, but maybe she'll watch the live stream later. So anyway, I'm going to skip to where we left off last night. For those of you who just want a quick review, we looked at 3 models of water, and the idea was you should be able to write a couple sentences about how is that model portraying the property of water. So this one was showing how water is less dense as ice, which is really weird because normally when things become more dense, they fall, right? Or they don't fall, they would sink in a liquid situation compared to their liquid selves. So water is a little bit different because those hydrogen bonds start to stabilize. So make sure you're able to explain why, and not just say like, oh, I know ice is less dense. Like I see it in my cup of water that ice is floating, or I can see it on the lake in the winter that ice is on top. Make sure you mention the structure of how when water molecules slow down, because when you have colder temperatures, remember kinetic energy is lower. And so the energy of motion is slow. And so these water molecules are slow. And so they actually have more stable bonds with one another, which actually spreads them out. And so if atoms are spread out in a given volume, remember density is mass over volume. So it's going to be less dense. So make sure you're able to explain that. Model 2 was looking at the polarity of a water molecule. So it was showing how those red arrows, I would say, is representing how oxygen is electronegative, and it pulls and unevenly shares the electrons between hydrogen and oxygen. That's what we call that's what polar covalent means, is it's covalent, they're sharing the electrons, but it's polar because oxygen is unevenly sharing it, which means, remember, electrons are negative, and so that makes oxygen act negative. It's partially negative. And so you would go on to talk about that in a free response, explain what you know about polarity, and then give maybe a biological example of when polarity is important. And so you could talk about basically every property of water is because it's a polar molecule. You could talk about hydrogen bonding, you could talk about cohesion or adhesion, you could talk about surface tension. All those things would be fair game and really good examples. And then model 1 was looking at, we've got 5 water molecules here, and the arrows are pointing to the bonds between them. So ideally you would have talked about hydrogen bonding. In all of your answers, make sure you're always defining what you're talking about. So if I mention hydrogen bonding, I better define what hydrogen bonding is and not just throw out that word. Oh, welcome everybody. We've got a couple new people here. Thank you for coming. We were just kind of reviewing where we left off yesterday. And these slides will be posted tomorrow once I get this video downloaded. Okay, so thanks for coming in. We're going to go over some more review questions, and then we'll have an open question time if you want. Okay, so number 3 on the study guide, and again, the study guide will be in a PDF form that you can download if you're a 5able Plus member. So number 3 says, without the presence of water externally and internally, organisms could not survive. Using properties of water in a biological example, explain water's ability to moderate temperature within living organisms and environments. Okay, so the bolded words that you'll see on AP tests are helping you focus on where to start, right? So you're going to talk about some property of water. Yes, good. I already have someone with one option that's correct. And that's what I also love. I talked about this yesterday, how with free response questions, a question like this, you could mention 3 or 4 things, and it's only asking you to look at one. So there's a huge variety of what you could say. So yeah, absolutely. I would talk about high specific heat is one really good property of water and how it can moderate temperature. So do you want to expand on that? What do you mean by it? When you say high specific heat, why does that help either an environment or an actual organism? Living in a coast? Yeah, Javier, absolutely. Yeah, so that helps total environments, right? We're talking about total ecosystems that are benefiting from the fact that, and for those of you who maybe are like, what's high specific heat? What in the world? High specific heat, yeah, and heat evaporation, good. So high specific heat has to do with water. It requires a lot of energy for it to change temperature, right? So I think of like the beaches here in Michigan. You know, we've got this hot sun all summer, and I walk out onto the sand in the middle of the afternoon, and the sand can burn your feet. It can actually burn your feet. It is so hot. But then the water, even though the sun has been beating down on it all day, from morning until night, the temperature itself is not changing that much. And so it creates a really nice, stable environment for the organisms that are living there. And then it's not just living organisms in the water, it's all the actual plant life, everything like that. Good, to moderate air temperature. Awesome, Diana. Yeah, so absolutely. So you can talk about stabilizing ocean temperatures, moderating air temperature. It's always cooler by the lake, they say, right? So if you don't live by water, trust the science of water and know that it is cooler if down by the lake or down by the ocean. Because of that moderation of temperature. Absolutely. Awesome. Really good job. And then heat vaporization, if you want to talk about that, it's very similar, but the idea is, okay, if I want to get water to go from liquid to gas, which is vaporization, that takes a lot of energy too. And so that comes into play once you start learning about organisms and homeostasis, and we talk about temperature regulation. This would be a good question that would be on an AP test because it's actually culminating lots of different units together. Good, sweating. Yes, Diana. So sweating, absolutely. The reason why sweating actually cools us off is that it takes so much energy for that little drop of water to evaporate that when it does evaporate, it actually is taking thermal energy with it. And so you feel cooler. Yeah, awesome. Good, because right, you have to break hydrogen bonds. That's going to take a lot of energy because water likes to be cohesive, right? Cohesion, it likes to stick together. And so that high heat of vaporization means it takes a lot of energy for it to go from liquid to gas, which is why sometimes when you have like a huge pot of water, if you're making like a huge thing of spaghetti, it takes forever to boil, right? Because water has a high heat of vaporization. You can't just turn on the burner and have it poof into boiling water. And that's also why if you think about on a humid day, when your sweat just kind of like sticks to you, why you feel so hot, because it's not actually vaporizing. It's not actually turning into a gas. So it makes you feel really hot. So good. I would talk about those things. That's excellent. I feel like you all feel pretty confident with that property of water, which I'm really impressed with. The only other thing that you could also talk about on this one is the ice property of water and how that can moderate temperature within an environment. And you could talk about ice and how it's less dense and that helps to insulate lakes below. Okay, so like here would be my 3 examples, but this is exactly what you all said as well. High heat of vaporization, talk about perspiration or sweating, high specific heat, moderating climates. I mean, you guys hit it, the nail on the head. You did great on this one. So really good job. You know your water stuff. Okay, next question, number 4. So we've got 2 amino acids, and if they were to undergo a dehydration synthesis reaction, what molecule would result? And then it would ask you to potentially circle the parts of the amino acid that will interact and draw the resulting molecule. And then also describe the type of bond afterwards. So again, yep, focus on the bold and think about what would happen. So I have amino acid 1, I've got the amine group over here, the carboxyl group here, I have my amine group here, and the carboxyl group here. So ideally, if you saw something like this, I'll just show you, this is my rough sketch, but I like to have my students, the hydroxyl group, is that what you're talking about? Yes, okay, good, the hydroxyl, yep. So the hydroxyl group, oh, it says I already sent that, huh? Oh, yes, I did. Okay. Yes, the hydroxyl group of 1 amino acid, right? The one that has the carboxylic acid section and then the hydrogen from the amine side. That's where your water's coming from. So normally I ask my students to circle like, hey, where's water actually coming from? Because a lot of kids will know, oh, I know that dehydration synthesis means we're dehydrating and synthesizing. We're putting 2 things together. But if they look at the actual molecule, they're like, I have no idea where they're coming together, right? If you do this, then you can know where that peptide bond is going to be. Because if I took away an OH from that carbon, remember carbon really likes to have 4 bonds, and it's left with 3, right? And so that's going to hook up to nitrogen that just lost its hydrogen. So that is where that peptide bond is coming from. That's your resulting growing amino acid. You could say when it asks for what molecule would result, yeah, you could call it like a growing protein. Definitely fill in peptide bond. That, remember we talked about that yesterday. It's just a specific word. P for protein, peptide, and primary structure, right? So the primary structure of your growing amino acid chain is connected by peptide bonds, and then draw the resulting molecule. So good. Great job. All right. Next question, number 5. Okay. So these are just a few pictures of, these are actual amino acids found in humans. If you take organic chemistry in college, you'll have to memorize all 20 human amino acids, and you'll have to know their 3-letter description. So like LYS for lysine, and you'll have to know their 1-letter abbreviation as well as the structure. And you really get into the organic chemistry of them. But anyway, these are just 4. And so the prompt says there's over 500 amino acids in nature. However, the human genetic code only codes for 20. Amino acids differ from one another in their R group. And 4 of these amino acids are shown below, or 4 of the R groups. Sorry, I didn't draw all the amino acid because that would have been a little ridiculous. So we're just looking at R groups, which is okay because the prompt says unique behavior and chemical interactions between amino acids that contribute to specific protein folding and function can be attributed in large part to the R groups. So we can organize R groups as polar, nonpolar, acidic, or basic. And when we say polar and nonpolar, that also goes hand in hand with hydrophobic and hydrophilic. So you might have had a teacher who focused more on those terms like hydrophobic, hydrophilic, but polar and nonpolar are attributed to the same thing. So this very first one, we have CH2 connected to a carbon with an oxygen and another oxygen. So think about, is it polar, nonpolar, acidic, basic? Yes, Javier, I would definitely call that polar. Yep, good. So it's polar for sure. Especially if you once you have an oxygen attached, that OH group can become polar as well. So the reason you can tell generally, oh shoot, I'm sorry. This one doesn't have a hydrogen. I would actually call that first one acidic. Sorry, I'm losing it. You can technically be acidic and polar, but so I would accept either one. But because of that true negative charge at the bottom, I would call it acidic because it has dropped off a hydrogen into solution. That's where that negative charge is coming from. But generally for something that's polar, someone just asked, how can you tell if it's polar? Is that if you think back to, hopefully your classroom has a periodic table that you can reference, but the idea is as you go to the right on the periodic table, you become more electronegative. So your oxygens are really electronegative, your nitrogens and carbons, not as much, but compared to something like hydrogen. So anytime you see something like an oxygen and a hydrogen together, you know there's going to be some sort of polarity because it's going to be unequal sharing of electrons. Mostly it's going to be between, oxygen, hydrogen, and carbon and hydrogen. Okay. Next one. Oops, I'm just giving you all the answers. Sorry. Yeah, so that first one I'd call acidic. There's actually no nonpolar ones. I meant to draw a nonpolar, but for some reason I made them all polar. I don't know why I did that. But a nonpolar one are usually your ones with just like methyl groups, like just CH3. Benzene rings are going to be nonpolar. So it's just like a carbon ring with no oxygens, no nitrogens. It's just carbons and hydrogens. And then basic is going to be positive because, again, it can pick up extra hydrogens if the solution or the environment is getting acidic. All right, and so if we start to think about this folding now, if I had an acidic over here and I had a basic amino acid over here, you can also just use your knowledge of positives and negatives and how they're going to interact and think about like, oh, well, this basic one on one side is going to interact with the acidic on the other. Your polars will stick together and they're going to also gravitate toward the outside of your amino acids of the primary structure because they like water, right? Just think about like polar molecule is going to be soluble in water, where something non-polar like a lipid is going to be hydrophobic. And it's not gonna want to be near water, which is the external environment. Okay, this is something you could reference as well if you're really struggling, but you just look up a chart of nonpolar versus polar and acidic and basic. Again, look at acidic and basic. They are true negatives over here and true positives over there. But that's nothing you'll have to memorize for AP Biology, okay? So don't worry about memorizing actual R groups. Here's something that's a little more applicable that might be a free response question. So 6 says the images below show a small section of the primary structure of an original protein that is fully functioning and a mutated protein. So we have threonine, proline, and then that middle amino acid, it's actually showing you the chemical structure so you can see how the mutation could affect behavior. So we had one with glutamic acid and then lysine, okay? The mutated protein still has threonine, proline, and lysine, but this middle amino acid has been mutated to valine, and valine looks like this. So the question says, using what you know about biochemistry, macromolecules, and protein folding, explain, A, how could this change occur? How could we have gone from a glutamic acid amino acid to valine? How could that happen? B, how would this change influence protein folding? And then C, make a claim whether or not this change would alter the protein's function. So go ahead and answer A first. How could this change happen? How would all of a sudden we have glutamic acid and then now it's valine? How could something like that happen? Oh yeah, Diana, you're getting really specific. Yeah. Diana is saying a deletion or insertion. Yeah, so we could have, when you're talking about deletion, you're talking about DNA, right? Which you probably have not gotten to this subject yet. But the idea is good, a mutation. Yeah, so absolutely. Normally, the only reason I would say it's unlikely that it's deletion or insertion, and this will make way more sense later once we get to transcription and translation, is that your fourth amino acid here is still lysine. And so I put that in there on purpose to show that it's just that one amino acid. And one amino acid is coded for by 3 bases. So like GAT or CAT or TTA or whatever. There's 3 bases that code for it. So if I delete one, that means I have 2 bases left, and I'm going to have to steal someone else's base, which is going to be lysines. And usually it's a frame shift then, and everything gets messed up. But you could still maybe argue and talk about the redundancy of the amino acid code. You could potentially argue, but you'd be strengthened in your argument by saying, oh, that normally insertion and deletion throws everything off, and it's a frame shift, and it will mess up the whole reading frame. So yes, Javier, that is really good. Yeah, these are the amino acids that, so this mutation is sickle cell anemia. Yeah, it's a really good classic example because it's literally just one little mutation and completely messes with the protein. Yeah, very nice. Way to identify your mutation. That's awesome. Yeah, so I would say you could keep it simple in your explanation for A. How could this change occur? A DNA mutation, right? Because you have to know, where is this amino acid coming from? Well, it was coded for by a gene, right? And genes are segments of DNA. So something went wrong there in that process of transcribing the DNA, or there was an actual mutation. B, how would it influence protein folding? On this one, I would want you to get really specific and talk about, okay, well, we went from having an acidic amino acid to now something that is hydrophobic, it's nonpolar, right? It's just a bunch of carbons and hydrogens. Think of it, remember, when you think of carbons and hydrogens, think back to the macromolecule of a fatty acid, and fatty acids make up lipids, right? Lipids don't like water. So we went from something that's acidic to something that is hydrophobic. And so if you mention that for B, how would it influence how it folds? Well, the insulator. Good. Yeah, so that section of the protein, I wouldn't say the cell is going to become hydrophobic. You would want to stick to the correct language and say that that section of the amino acid is, rather than being on the outside, is now going to want to try to huddle toward the inside, and it's going to maybe pull the ones next to it toward the middle as well. One tiny indentation that's incorrect on a protein completely obliterates what it's supposed to do. Yeah, so you could say it probably would fold more, right? But the key would be to show that you would know that something hydrophobic means it's going to move toward the center of the protein. It's going to try to be away from the liquid environment that all life lives in, right? So these amino acids, and this again, as we build on our knowledge in AP biology, you'll have so much more stuff to be able to pull from. Like right now, we're just looking at amino acids, and we don't have the full picture of, well, where is this happening, right? Where is this amino acid being built? It's being built in the cytoplasm, okay? And that's a aqueous environment. And so it'll all come together more, and you'll have a lot more to say in your explanations once you can pull from concrete examples of stuff that you understand. And then C, make a claim whether or not this would alter the protein's function. Sometimes with making claims, as long as you have good evidence to back up what you say, there could be a couple claims made. The more obvious one would be, we've been kind of really hitting it hard, at least in my class, that shape of a protein is everything. Right? The shape is everything. And yeah, Ida, absolutely. So because structure affects function, right? So I feel like this question's almost baiting you to agree and make a claim that says, yes, of course this is going to affect a function because we have an amino acid that's acidic and now it's hydrophobic. You could absolutely talk about it being inactive, Javier. Absolutely. This would not be denaturation. No. So denaturation means something has happened in the environment that has made a normally functioning protein fall apart. So think it's denature. So we're taking it away from its natural state. So that would be if you were to heat some protein up to a super high temperature, those bonds will start to break and it completely unravels and it no longer has its function. So this would not be denaturation. This would just be improper folding. The protein would never have made its full functioning structure because of its incorrect amino acid. But yeah, denaturation is more like if you guys did an enzyme lab maybe in class, if you did like a catalase enzyme lab where you maybe heated it up or you added lots of salt to it or you, what are some other things? Yeah, if you made it really acidic or you change the environment, usually it's temperature that causes denaturation. Is this a change in the primary structure? Yeah, so Ida, this mutation has caused a change in the primary structure. So the primary structure is just that initial linear connection of amino acids all together. So we went, this is only just showing 4. Yeah, I can go over part A, sure. So what this question is showing is just a snapshot of a protein. It's just showing 4 amino acids. The original protein has threonine, proline, glutamic acid, and lysine. So those 4. And the reason I have glutamic acid actually written out with all my carbons and hydrogens and all that is so that you can see the structure of what glutamic acid is, because you don't have them memorized. We don't know just off the top of our heads that proline is hydrophobic or whatever, right? So that's why I have just that section wrote out. Anyway, so our original protein has these 4 amino acids. The one that obviously I'm wanting you to look at with the orange box over it, it's glutamic acid. So it's an acid R group, where now we have a mutation, a mutated protein. It has 3 of the 4 amino acids, but now instead of a glutamic acid, valine is in its place. And just looking at it, you've got carbons and hydrogens together. And I told everybody, I said, hey, remember that's kind of like a fatty acid. I mean, it's not an actual fatty acid, but when you see carbons like CH3 and CH and all that, like you should be thinking nonpolar. This is going to be something that's hydrophobic. So part A, Ari asked about how could this change have occurred? Ideally, you would talk about there's something that went wrong with DNA. So either there was a DNA mutation that now is probably a substitution, right? So like a C became a T. And so now that actual code, and again, this will make a lot more sense. And you'll have a lot more to say about it once you get into your DNA unit and if you get into protein synthesis more. But the idea is DNA codes for protein. So if there's a misstep protein, if my protein is actually wrong, I can trace it back to DNA. So something happened with the DNA and now my protein is going to be mutated. Most mutations we don't even know happened because you could have 5 different sets of codes that all give you the same amino acid. So most mutations are not actually bad. We don't even notice they're happening. The cells don't even know. Proteins are still made. Everything's good. But obviously with certain diseases, like Javier mentioned, this is an example of sickle cell, where one DNA mutation causes an amino acid change. And because these 2 amino acids are so different, you have an acidic one and a nonpolar one, it's going to change how the protein starts to fold. So you start out with that primary structure. They're all like beads on a string, kind of. And then they start to interact, right? Because it's chemistry, okay? So if you have something that's positive and negative, they're going to come toward each other. If you have 2 positive things, they're going to try to get away from each other. If you have something that's hydrophobic, like valine is, amino acids are in this aqueous cytoplasm. They don't the hydrophobic things don't want to be near that. And so they all kind of huddle toward the middle. And so you're going to get a completely different protein shape. And if we've learned anything about proteins in the macromolecule live streams or macromolecules that you've learned about in class, structure is everything, right? So the shape of a protein dictates whether or not it can do its job. If its shape is altered, you don't have your protein doing its job. Okay. And then I know some of you were talking about denaturation. This would not be denaturation because the protein's already mutated. Denaturing has the word nature in it. So think the protein was made correctly, but then something happened in the environment. Like I get really sick and I have, hey Amanda, what's up? So if I get super sick and I have a very high fever, right? After a while, your doctor would want to know, like, have you had a high fever for 24 hours, 48 hours? What's going on? Because those high temperatures will start to denature and unfold your proteins for things like enzymes in your body that need to do their jobs. Yeah, and Javier, absolutely. Salt solutions too can totally denature. You put in some salts, positives and negatives are going to fight for those acidic and basic R groups. And absolutely, that'll start to pull apart your protein as well. So great job. Okay, any other questions on this one before we move on to number, where are we? Number 7? I think we're good. Okay, this one's a little bit more simple because it's just multiple choice, but take a look at that little cartoon. Number 7 says, based on your knowledge of the polarity of water molecules, the solute below is most likely blank. So A, positively charged, B, negatively charged, C, without charge, D, hydrophobic, or E, nonpolar. I realize that I have 2 sets of letters there. I don't know what happened. So this is part multiple choice, part short answer. You'd want to be able to explain. So the solute is the little black dot in the middle. You had a question similar on your unit test? That's funny. Yeah, so the black dot is the solute, and if we're talking about water molecules, you can assume that the purple and orange molecule is water. And so if you have these water molecules with their purple ends all coming in on the solute, think about what you must know, what must be true about that molecule in the middle. And why? How would you know that? How could you explain it? All right, Javier and Diana are saying A, it's positively charged. How do you know that? Thanks. It's positive because oxygen is slightly negative in water. Good. So, right. So, first of all, we're assuming the purple part is our oxygen. Good. We positively charged to bond. Awesome. Nice, Diana. Yeah. So, Javier, I don't know what term you're maybe thinking of, the polarity of water you could mention. You could say, you know, water's a polar. Oh, hydration shell. Nice. Yeah, you could definitely talk about that. You could just talk about polarity of water and say, you know, these molecules on the outside are representing water. I know that water is H2O. It's polar. Here's what polar means. Talk about that for a second. About those unevenly shared electrons. Good. Like Diana said, it would be positively charged. The solute would be. And so it's interacting with the partially negative oxygens. In a real free response on the AP test, be really careful about terminology. If you start to just call oxygen negative, you might get into some trouble, right? Because we're not talking about an ionic bond between oxygen and hydrogen. So they're partial charges, or you could say slightly negative. Yeah, awesome. So slightly negative. So just those little things that even though you know what I know you know what you're talking about, but if you lose sight of those tiny details, it doesn't show that you truly understand polarity and the idea of partial charges. So just a little tip there, because sometimes, especially on a question you feel confident about, it's easy to just kind of right, and just spill everything you know, but just be careful about what you say, and make sure you show them exactly what you know. But really good. That's a good sample question for everybody. Okay. I have, I think, one or two more left. So number 8 says, imagine the amino acids below join to form a small polypeptide chain along with several others not pictured below. So just like that other mutation problem, we're working with 4 amino acids just for simplicity purposes, but they're not the only ones on the structure. We could have like a whole, let's say we've got 40 amino acids. Okay, so we have 4 amino acids. A says, which amino acids could form a hydrogen bond with another amino acid in the chain to stabilize the secondary structure of a beta-pleated sheet? So just think about A and think about which ones could form hydrogen bonds with other amino acids in the chain. I'll give you a hint. This one does not have to do with R groups. Yes, yeah. Javier says, oh, yeah. So this is it's almost a trick question because we hit R groups so hard in understanding how they're going to behave, but we forget that each amino acid has an amine group and this carboxylic acid group. Okay, so once you have those all aligned in a string and they can start to fold in on one another, your nitrogen and carbon, like side groups there, those are actually what can form your hydrogen bonds and give you that, secondary structure or second level of folding, right? So our primary structure is just peptide bonds. Secondary structure is hydrogen bonds. Some proteins have beta-pleated sheets where it's like these nice, it looks like an accordion or like a fan that you could make yourself out of paper, or they get these really nice alpha helix. This question is just talking about the beta-pleated sheet. But yeah, absolutely. All of them would be able to start to interact and have that secondary structure of hydrogen bonds. Good job. B, which amino acid could form? So all secondary structures can form hydrogen bonds. Yeah, so that is basically what the secondary structure, it's kind of the second tier of protein folding. It's, protein folding doesn't just happen, right? You've got your primary structure that's made during protein synthesis, and you have to get to all these different levels before your protein is functional. So this is like on your way to becoming a functional protein. So you get these sections of your amino acid that starts to fold in on itself, and they're all hydrogen bonds for secondary structures. It's a good question. And then B, I'm just going to show you a picture and then we'll go back to B. So here it's talking about your alpha and your beta, alpha helix and your beta sheets that can happen. And so I know it's hard to see, but the little purple circles are your amino acids, and the dotted lines are just between each of them to show that it's not just the R group, it's the actual amino acid itself. It's the nitrogen and carbon and oxygen between them all that are interacting for that secondary structure. Okay, B might be a little easier if you are really comfortable with R groups, but B says, which amino acid could form disulfide bonds with another amino acid to stabilize the tertiary structure? Oh, you guys are so fast. Good. Awesome. Yeah, 4. You guys are all agreeing. Yep, number 4. You see that sulfur right in there, a disulfide bond. You would either need another amino acid that looks just like that somewhere down the line. That really helps to stabilize that tertiary structure of an amino acid. So, right, without that particular amino acid, you would not have a disulfide bond, but that really helps get that third layer of tertiary structures going. Something I just want to clear up before we move on. Your secondary structure has lots of hydrogen bonds, but On top of that, we also have hydrophobic and hydrophilic folding as well. So don't think of it as secondary structures, it's just hydrogen bonds. That's a big part of it, but the secondary structures also start to attribute to hydrophilic and hydrophobic components. Okay. Awesome. Cool. Actually, let's do if I were to add a C to this, like part C, which amino acid would participate in hydrophobic interactions? Which ones would have a hydrophobic interaction? Or which one would be hydrophobic and want to hide in the middle? Yeah, Javier, you're killing it. Awesome. Yeah, number 1. Yep, and that's the benzene ring I talked to you about earlier. It's just carbons and hydrogens, that's it. Carbons double bonded to carbons, and hydrogens, they're not pictured, but they are there. Absolutely. Number 1, hydrophobic. Good. Great. Okay, that's actually all I have. Oh, wow, we went 48 minutes. Wow. Time flies. Is there anything